Dr. Shakira?
She might.
Oh, she's mute again.
Yeah. I made her a co-host.
Okay,
so
now I
she,
can.
yeah.
Can you hear me?
Yeah.
Okay, cool. Sorry about that.
Hi, Dr.
problem.
Shakira. How are you?
Alhamdulillah, I'm doing well. Thank you.
Alhamdulillah. Thanks. Thank you to join us again, Dr. Shakira. السلام عليكم جميعا الأساتذة الكرام. هذا اليوم إن شاء الله برشتنا الافتراضية الموسومة Optimization and Quantification of Immunofluorescence Signals in Virology Research.
Can I start or no, not yet.
So إن شاء الله اليوم guest speaker دكتور شاكيرا أدوتنجي من نيجيريا. وللذين يعني ما التقينا ما جو بالورشة العمل السابقة، دكتورة شاكيرا أدوتنجي من نيجيريا، هي طبيبة بيطرية وكملت ماجستير ودكتوراه في جامعة تكساس A&M يونيفرسيتي في ولاية تكساس، وبعد ذلك سوت بوست دوك في جامعة كانساس ستيت، وهي الآن طالبة بورد في علم الأمراض البيطرية. So this is a short introduction about Dr. Shakira Adetunji. She is a resident anatomic pathologist at Kansas State University, where she is involved in performing autopsy and biopsy service on a wide variety of animal species, as well as teaching second year and final year veterinary students, general and systemic pathology, respectively. She completed her master and PhD at Texas A&M University. Her dissertation focused on understanding the immune pathology mechanism of brucella-induced pregnancy complications using mouse model. Dr. Shakira was postdoc fellow working in collaboration with the USDA and Kansas State University on foreign animal arthropod-borne diseases, specifically Japanese encephalitis virus using pig models. Dr. Shakira, thank you to join us. I know you are super busy, and let's get started, inshallah.
Okay. Thank you very much for the introduction. Like Dr. Mar said, my name is Shakira, and today I will be talking about how you can optimize and quantify immunofluorescence signals, especially when it's related to virology research. So a little bit of introduction about me. I'm originally from Nigeria, and that's where I got my DVM. And then after that, I went to the US and I got my master's and my PhD at Texas A&M University, then moved to Kansas where I did my postdoc, and I'm currently completing my residency program in anatomic pathology. So the outline for today's presentation is as follows. I will talk a little bit about the basics of immunofluorescence assay, the procedures, the protocol that you can use and how you can troubleshoot, the different materials that you can use and how you can help yourself to get acquainted with the different methods that there are out there for IFA. And after that, I will talk about how the possible ways that you can analyze your data through qualification of the signals and quantification. The several applications that you can use IFA for, and particularly I'm going to be talking about one of the papers that we published using this protocol in virology research. I'll let you know the experimental design, how we came about the methods, results, and then conclusions. So the basics, IFA. From now, I'll refer to immunofluorescence assay as IFA, okay? So just like I talked about the other time where I talked about flow cytometry, it's basically the same kind of process, okay? Antigen, antibody interaction, and then you get your signals. So basically, you identify and characterize the different structures that are either within the cell or on the plasma membrane, like that one, or within the nucleus. So you can use IFA to qualify and quantify different structures of protein expression in a cell. And like I mentioned before, it's antigen-antibody interaction. It's just a basic step, okay? And here, because we're talking about fluorescence, your antibody has to be conjugated with a fluorophore or a fluorochrome. And then afterwards, then you can try to analyze your signals by seeing the signal intensity, that's a qualification, and then quantification, how you can interpret that signal into statistics that you can use to like analyze or interpret your data. Okay, there are two basic methods, the direct and indirect methods. For the direct method, you have your cell of interest, and then you have a particular protein structure that's expressed by the cell, either within the cell or on the plasma membrane. And then you get your antibody that reacts against that epitope of the antigen. And the antibody will be fluorochrome conjugated, and that's how you detect your signal, okay? For the indirect method, you have the same cell with the same protein structure you're trying to characterize. You have the primary antibody. However, that antibody is not conjugated, okay? And then you have your secondary antibody that's conjugated and will help you see your signals. The difference between these two methods is that direct method is kind of expensive. It's faster, okay? You just add your antibodies and boom, you're done. It's faster, but it's not as complicated, but it's expensive because that means all the antibodies you use has to be fluorochrome conjugated. And there's very few out there, especially if you're working with proteins that are kind of rare or in a rare animal species. For the indirect method, you have your primary antibody, doesn't have to be conjugated, so you can use several antibodies. And then it's kind of longer, but it's the preferred method if you're thinking about cost, okay? So another step that is important is here. Let me see if, okay. Oh, sorry. Is with the direct or indirect antibody step. So let's take, for example, this is one of the steps that can help you troubleshoot your protocol. Let's take, for example, these cells are from mice, right? And what you're trying to quantify is the expression of CD4, which is an immune receptor on a T cell. Let's say these are lymphocytes from a mouse, right? And you have expression of CD4, okay? So you're gonna get a primary antibody that's fluorochrome conjugated, recognizes CD4, and then you have your anti-mouse. So if you're trying to look for antibodies, okay, you wanna go online and look for anti-mouse CD4 because you're working with a mouse, okay? So you're gonna look for antibody that's that reacts with CD4 antigen in a mouse. And then with this step, you're done. With the secondary antibody, it's kind of a little bit, you need to pay more attention with the kind of antibody that you get. This is also a lymphocyte, for example, from a mouse, and you have this epitope, CD4, right? You have your primary antibody, however, it doesn't have to be conjugated. But your secondary antibody has to be the antibody in which it can recognize mouse proteins, okay? So this one will be anti-mouse CD4, but your secondary antibody has to be something that recognizes mouse. Okay, so that's goat anti-mouse. It could be from rabbits, it could be for donkey. So what you're trying to look for is any antibody that's raised in something else, but recognizes a mouse protein. This process can be complicated because even if you get the first step right, and you chose something different, for example, just kind of like any kind of antibody that does not even recognize mouse, even though this bound to your protein, you're not going to see signals because what this one is looking for is anything that looks like a mouse. All right. So that's where you have to pay attention while between differentiating direct from indirect process, okay, of the IFA. So for immunofluorescence assay, there are different controls that you should consider. Positive control, okay? Positive control is a tissue that you know for sure is gonna express the protein that you're looking for. For example, if it's something that you're not sure of or something, you can use a stimulator to stimulate the expression of that molecule in your cell and use that as your positive control. For example, if it's Brucella that you're trying to look for, you have to be sure that those cells are already infected with Brucella, right? So you have a control like that to be sure that your protocol is working. Your negative control is the cells, the population of cells that you know are not going to express that protein. The no primary antibody control is the one that you're trying to see if your secondary antibody is having some background reaction to the cells itself. Okay, so that's needed if you're doing the indirect method of the steps, of the protocol steps. The absorption control, it has to do with being sure that your antibody is actually reacting to the specific epitope of antigen of interest, okay? So there has to be an extra step in your protocol where you react, put your antibody together with the antigen. That way, those antibodies bind to antigen in the solution, and there is no antibody available to actually bind to your cells of interest. Therefore, you wouldn't see any signal. That's a control that will help you understand that your antibody is actually specific and reacting to your immunogen of interest. So all of these have to be taken into consideration while you're troubleshooting your protocol. So let me go stepwise on how you can design an IFA protocol. You have your cell culture, right? So here I'm using cell culture as an example. You can also do IFA on tissue samples, okay? But this is cell culture. And that's because that's the basis of the paper that I'm going to present much later. You have your cell culture, okay? You have to prepare the cells for counting before you even think about starting your experiment. You've been growing the cells for a while, and when you're ready to do your assay, you have to know the number of cells, right? So you decant the media, the old media, and put the cells within a tube. You can use trypan blue and then count your cells. The counting, there are a lot of like automated cell counters out there, and you can also use this clicky thing for quantification of the cells. Once you know the number of cells, that would help you because further down the line, you're going to infect the cells with virus. So you have to know the amount of virus that you need. For example, we call it MOI of infection, that's multiplicity of infection, to help your quantification later down the line. So after counting the number of cells, then you plate the cells in different wells. It depends on what kind of assay you're trying to do. You can use 24 wells, you can use 2 wells, which I will also show the protocol for that. So after plating the cells within the wells, you infect the cells with your virus. You incubate for however long, depending on the virus that you're working with. And then the next step is kind of critical, okay? Because it would vary depending on what kind of protein you're trying to quantify. If your protein is on the cell surface, like CD4, for example, you don't need to fix the cells. You can just incubate with the primary antibody and move on. However, if your protein of interest is inside the cell, you have to fix the cells and permeabilize the cells so that the fixation reagent will help you poke holes on the cell membrane so that your antibody will be able to enter the cells and bind to the antigen of interest, okay? If you make a mistake in this step, you forgot to fix or permeabilize or anything like that, you're not gonna see signals. Why? Just because your antigen of interest is inside the cell. If you're looking for antigens on the surface of the cell, that's okay, because you don't need to fix. Your epitopes are there on the cell membrane, okay? And then after that, you incubate with antibodies if you're using the direct step or the indirect step, primary or secondary antibody. And then you can use a fluorescence microscope and detect your signals. There are different fixation methods that you can use. The quicker and faster one is to buy kits by different manufacturers like Abcam, Thermo Scientific, and eBioscience, and other manufacturers like that. They have a kit which is just like straightforward. The kit has the reagents for both fixation and permeabilization of the cells, and then you just use that. Some people use their own methods of forming their own paraformaldehyde and use that to fix the cells. And then some people also use methanol. So you can use different fixation and permeabilization methods. However, the quick and faster one will be fixation with the manufacturer's kits. They already have all the combinations in there. You just use it and then you move on. So I was talking about the different methods that you can use, the different kinds of plates or materials that you can use. Depending on your assay or your protocol, or even your virus, you can decide which or the volume of your experiments. You can decide what kind of plates you want to use. EBD, IBIDI, they're using this slide method. They have this in stock, and I've used this before as well. So if they have this in such a way that you don't even have to like transfer the cell at different points, you know, so you can start your culture inside these wells right from the very beginning. You start your culture, and that's where you finish all the protocol that I mentioned earlier. So depending on the virus, the volume, and the number of cells that you need to plate, then you can use these wells, 2 wells, 4 wells, 8 wells, and then you can also use 24 well plates. I use this one a lot because I was dealing with different volumes of different kinds of cells using several cell lines at the same time. If you're in that kind of situation, you can use a 24 well plate where you have, so that way you'll be able to even have your controls on the same plate, which is always easier, okay? So this is kind of the method. You seed your cells, you let them grow for however long, depending on the cell line. You fix and permeabilize the cell, you stain the cell, and then you take this same slide into your fluorescence microscope directly, okay? But at some point before you image, you have to clip off this box of wells. However, you still have the demarcation of those different wells on your slide. I also used Keyence microscope. This is how it is. This is the whole thing, all right? You can use this one for even for H&E. You can use it for IFA. So when you're done with the last step, when you're ready for your imaging, you pull up this cover and then you just insert your slide or plate. So it has, depending on what you want to use, it has different shapes for plates, for slides, and things, and trays for slides too. So you just put your cells in and then you click on this, and every other thing you're going to do will be on the computer. So that was what I used for my experiment. And the good thing about this is also that once you're done with your imaging, it gives you like the spreadsheet of numbers that you can put together and use statistical analysis for you to quantify your signals. So now you're seeing the signals, but how much signals are you seeing? How many cells are expressing that? So that's why this is really good. When you have all of that information, you're able to, plot a graph to show your audience the trend or the tracks that the thing is going through. So this is an example of one of my results from using, I used this plate, and then I used this microscope, and this is what I got. So now, depending on how your experiment is, which I always tell people, make sure that your cells are confluent before you start infecting, okay? And depending on how long you need to incubate your primary antibody or secondary antibody, there are times that I had to incubate overnight my primary antibody. And if you're incubating overnight, you have to take that into consideration about your cell multiplication. Like, would it be like more cells in 24, 48, 72 hours. And you would also take into consideration how long you've been like incubating the virus, your virus with the cells. Here you have confluent cells, okay? And after my staining, primary antibody, secondary antibody, and everything, you can see my signals. So they're inside the cell. That's what I was looking for. So I had to fix and permeabilize the cells so that my antibody can go into the cells and bind my antigen of interest, okay? This is what you get when you don't have a confluent cell. But here I use a very high MOI, multiplicity of infection. So most of the cells got infected. Here I used, a reduced MOI, and you can see just a few cells got infected, right? So here's what I meant when I talked about signal quantification. When you use the Keons, I'm not in any way advertising for Keons. It's a really great one. So that's why I'm saying it here, okay? It will give you this spreadsheet of numbers. And at first you can get overwhelmed by all this number, like, oh my God, what's going on? Which one am I going to use? They're really helpful. I called them several times and they helped me through this during my process. So you're going to have some numbers like count. That's the total number of cells expressing your signal. Average brightness integration, like average total brightness of each cell. And you can also do brightness integration of all the cells in that field. And then you can do average, like the mean, the median, and things like that. And you can use that to produce graphs like this to tell you how many of your cells are positive for the antigen and what's the fluorescence intensity, how bright are the signals. In other words, how much of the protein is being produced in the cell to give you the fluorescence intensity. So I'm going to talk a little bit about one of the papers that we published using this protocol. It's infection of several porcine cell lines with Japanese encephalitis virus. So like I always do for all experiments, for all my presentations, is that you have this outline of what am I going to do with my experiment? What's my end goal first? And then you backtrack and say, how do I get to that goal, right? So the very first step is going through the background, your literature search, and be like, what's out there in the literature about my methods? And then what question do I want to answer? What methods? How am I going to answer the questions? What methods can I use? What are the results that are expected? And of course, for every experiment, you're going to have limitations and lots of troubleshooting, and that's okay. And then you get your results and conclusions, okay? So I'm going to start with background and rationale. Like I said, Japanese encephalitis virus, which I'll refer to as JEV from now, it's mosquito-borne virus, it's zoonotic. You can have up to 30% case fatality rate in infected people. It's highly endemic in the Asian Pacific regions, and you can have natural transmission cycle between mosquitoes, birds, and pigs. And I'm using pigs here because pigs are really important in the transmission cycle, okay? So look at this diagram where you have a mosquito that feeds on amplifying host. We call them amplifying hosts because they have a very high level of viral amplification, okay? The mosquito feeds on birds, and then the cycle continues. And the mosquito can get out of the cycle and infect dead-end hosts like humans and horses. Like I told you, it's zoonotic, right? So it's a big problem. You have mosquitoes, you have pigs, which is ubiquitous, it's everywhere. And you can have the continuous transmission of the virus in this cycle. So what are the manifestations of JEV? You have neonates, which can have up to 100% mortality rate. It presents mainly as neurologic disease in neonates. In adults, not as much. The only complication we have is in the reproductive system. And mortality is rare. It's just you're going to have abortions and several reproductive complications, okay? And this is why it's important for you to look at the literature and see what's out there before you even start thinking about your experiment. The tropism of the virus and pathogenicity of JEV is severe in several systems, right? You have the respiratory system. And that way, if your goal of your research is to do a research on respiratory pathogenicity of the virus, and you want to start with cell lines, your best bet will be using cell lines from nasal turbinates. If it's the innate immune system, macrophages are the big guys in that field. And then reproductive system, like I told you, it causes reproductive complications in adults. Your best cell lines will be testis cell line or uterus cell lines. The urinary system too can be affected, so you can use for like kidney, renal epithelial cell lines, okay? And then research question, right? What question are you trying to answer? And that's why it's important for you to go into the literature. Maybe your question has been answered in some way before. So you want to be sure so that you're not just like being redundant in the contributing to the literature. You want to do something novel, something novel that can help drive and improve what's being done in the field. So what's our rationale? Our rationale for the experiment is that, well, we know that it's still like pending. We don't really have all the information or the total full understanding of the viral replication cycle, okay? And the host virus interactions. We still don't know. There's several studies out there, but it's not been fully established. So the objective was to develop an in vitro system for us to see the early events of JEV infection and replication and the host response, okay? So our hypothesis was that several cell lines will be susceptible to the JEV strain. So we use the attenuated strain here, okay? So like I always say, every research needs to have a hypothesis. What question are you trying to answer? So it's a way of formulating like your research question, what you're gonna do. Like I told you, JEV affects the respiratory system, it affects the kidney, testis. So we use the nasal turbinate cell line, the kidney cell line, testis, and then macrophages, of course, okay? Methods, so how did we do this? Like I said, let's say we have cryovials of cells, frozen cells, to jumpstart the experiment. You get your cryovials, and then you put them in the media. Depending on the cell, like I told you, I use several cell lines. So every cell line is different in how long they need to be incubated before they jump back to life, okay? So you have to take that into consideration as well, so that when you're planning your IFA, you have all your cells ready, all your cell lines ready, especially if you're doing several cell lines, and then you prepare them for infection, okay? And then let's say I have all my cell lines, and then I have my infection with MOI1, you calculate your MOI before you start, and then you infect the cells, and you incubate for different hours post-infection. Here, because it's a virus, you know, you do your plaque assay to see the cytopathic effect of the virus, and then you can count how many cells are still alive after each time point. So you can use Trypan Blue for that. You see the live and dead cell ratio. And then if you're going to do IFA, which is the main point of this conference, is IFA. So then you also have to take into consideration at what time point is best for you to start your IFA in which you start seeing signals. Is it at 24 hours post-infection? So you're going to have to do all those troubleshooting and then pick the peak period where you know that you have a lot of virion particles inside the cell, so you can see a lot of signals. So I put this plate here just to show you that you don't have to use the fancy plates and slides that I showed earlier when I was troubleshooting, because those are really expensive, okay? But when I was troubleshooting, I used the regular 24-well tissue culture plates to do my troubleshooting of antibody incubation and all of that. Once I established my protocol, then I moved on to the fancy plates for, you know, fancy pictures and to make my life easier, sort of. So, results. I will start with the changes that I saw with viral infection before I move on to IFA, okay? So these are all the cell lines that we use. We used to BHK kidney cells, macrophages, the nasal turbinates, the kidney cell line, and the testis cell line, okay? So these are the changes, the cytopathic effects we're seeing CPE at 0, 48, and 98 hours post-infection. BHKs are kind of like my positive control. Remember earlier in this presentation, I talked about positive control, okay? They're kind of my positive control because they tolerate, they're highly susceptible to JEV virus. And you can even tell that at 0 hour post-infection, the cells are nice and spread out. At 48 hours post-infection, they're beginning to be rounded and really, really dense like that. And by 96 hours, you're not even seeing a lot of live cells anymore, okay? So that's my positive control. And these are the cell lines that I was testing. So we use CD2 porcine macrophage cell line, and you can see that the CPE is most severe at like 96 hours post-infection. And for the other cell lines, you're not really seeing a lot of CPE at 48 hours post-infection, and mostly at 96 hours post-infection. And this, like I told you, you need a control cell, a negative control. My negative control here was that we had all these cell lines and then they were not infected with JEV. Okay, so you can still see the cells even after several hours post-infection. Okay. So here is what will help you determine if what time point you're gonna do IFA. Okay, so here is showing the virus titer from 0 to 120 hours post-infection. My positive control cells, BHK, they go high, and at 48 hours, that's the peak of virus replication, and then they begin to die, right? The same for CD2, the peak period was 48 hours post-infection with the kidney too. However, for swine testis, the replication continued throughout 120 hours post-infection. Okay, so here, if I want to focus on the porcine macrophage cell line, the time point that I'm going to choose from my IFA will be 48 hours post-infection, because that's the peak time where you have a very high level of virus titer. Okay, so if I'm trying to quantify the signals of JEV proteins within the cells of macrophages, and I have this graph, my best bet will be that at 48 hours post-infection. So that's how you begin to think about your next steps and the next questions you're going to answer for your experiment. Okay, here we're looking at the different, the survival of cells after the different hours post-infection. Okay, this is BHK21, my positive control that I know are highly susceptible to JEV virus. Okay, at this time point post-infection, they're pretty much gone, right? So, but here with the porcine macrophage and the other cell lines, I still have them surviving even up to 120 hours post-infection. Look at the survival time for the testis, for the swine testis, for the nasal turbinate cells, and compared to my positive control BHK. Okay, so let's talk about the IFA protocol for this one. So the same process. I get my cell culture, you do the infection, you make sure that you count your cells, and then you determine your MOI with that. The reason why you want to count your cells is that if you're going to go far out, up to 120 hours post-infection, the number of cells that you're going to plate will be smaller because you want to leave room for growth, okay? So if you start with the highest number, they may not last up until 120, and they may be dying, not because of the virus, but because there's no room for them to grow, okay? So those are the kind of things that you take into consideration during your troubleshooting. So this is infection of the different cell lines with JEV. I have my DAPI, which sends the nuclei, my JEV NS1 antibody, which sends the NS1 protein within the cell, and this is when they're merged together. Here is my positive control. See the amount of signals that I'm seeing with BHK, infection of JEV, and then you see this small, relatively lower number of signals with the other cell lines, and this is when they're merged together. And of course, you just don't want to show the qualification, the quality of your signal. You also want to help your readers or your audience understand the signals, and that's where quantification comes in, okay? Here, the quantification, you're quantifying the number of cells or the percentage of positive cells for the NS1 protein. Here's my positive control, okay? If I wasn't seeing anything here, then there's something wrong from the very beginning of my experiment. That's why you always have to have controls in your experiment, okay? And then this is the fluorescence intensity, which means how intense are the signals that we're seeing, which correlates with the amount of proteins or amount of viral particles within the cytoplasm of the cell. So conclusions from the experiment and from, so here I'm showing other, a bigger picture of your signals within the cell, intracellular, okay? If what I was saying was like inside the nuclei, like inside these blue cells, then I know there's something wrong with my experiments because the protein I'm interested in is inside the cell. If the signals I'm seeing are on the plasma membrane, then there is a problem because I'm not looking for plasma membrane proteins. I'm looking for proteins inside the cell. So those are the kind of things that you use to interpret even the papers that are published out there. You want to know what the protein they're trying to characterize. Is it intracellular? Is it the plasma membrane? Is it within the cell nuclei? So here I'm just showing how typical signals are within the cell. Oh, yeah. And with that, that's short and sweet presentation that I have. And I would like to acknowledge my PI and my colleagues at the USDA for really helping and funding this project. Dr. Senicharo and the collaboration between Kansas State University Core Facility, the Center for Outcomes and Research Epidemiology. And I will take questions.
Thank you, Dr. Shakira, for this nice presentation, and congratulations for the publication about the encephalitis virus. Please, any question to Dr. Shakira?
Please feel free to raise your hand, and so we will be able to unmute you. Thank you.
From Dr. Dali, I guess. Dr. Sarhat, I think Dr. Dali, she has a question.
Did she raise your hand?
No, she wrote something in the chat. I have one question.
Okay.
Dr. Dali Ahmed.
Which one?
Dr. Dali Ahmed.
Oh, Dr. Dali. Okay.
Dali, yes. Thank you.
تفضلي دكتورة داليا.
Okay. Assalamu alaikum, Dr. Shakira and all of the colleagues. Can you hear me, Dr. Shakira?
Yes, I can.
First of all, I want to thank you about the simply presentation for IFA, and it is was rich with information. So I have one question about the optimization of the IFA. Okay, so you mentioned earlier about the numbers of cells. We need to know the exact number, or not exactly. We need to calculate the numbers, right, of cells before we start the experiment. In this case, you use, as I remember, 5 types of cells, 4 or 5 types of cells, right?
Yeah,
so all these cells have same numbers?
No.
No. Yeah, this is my question. So it is depend on the multiplication rate of each type of cell, right?
Exactly. Yeah.
Exactly. So you need to know the peak of each numbers you need to do your experiment. Am I right?
Yes, you are.
Can you explain more about this? For example, the macrophages is not so multiplication rate of the macrophages is not the same for kidney cell.
Right.
Okay, so how you did all these experiments for different types of the cells, and definitely you need to do dilution for the primary antibodies, right?
Yeah.
This is the second one. So the second question. So the dilution of the antibodies was same for all these type of cells?
Yes.
Okay. So can you explain that, please?
Yeah, so thank you. Let me start with the cell number. And can I go back? Let's see. Okay. So before you even, let's go to. Okay. So before you even start infection or IFA or anything, you need to establish your cell numbers,
Right.
the timing and everything before you even start infection for each cell line. So that could take you several weeks in trying to, get to that point where you know that, okay, BHK really grows really fast. Okay. So
Right.
at what point would I stop them, or would I infect so I don't just go through infecting them when they're overgrown? So that's the very first step that you need to establish for all the cell lines. They have different shapes, they have different media, they have different timing that they need to grow to their maximum, sort of. So what you need to do first is plate them without infection or anything.
Right.
Forget this one. Without infection or anything, plate them.
And just
And
count
see-
them. Okay.
Yeah, so how many? So once you're ready to, let's go here. Here. Once you're ready to plate them, you count them, right? You also need to know how many cells you need to plate. It's different for 24 well plates, 12 well plates, 8 well plates. Okay. So I think on Thermo Fisher website, you're gonna have the number of cells that can fit into a well at a particular point in time. They'll tell you the area of this well is XXX centimeters square or something.
Yes.
And the maximum number of cells that you can fit in it is maybe 1 million cells. That way, if you want to start plating, you're not going to start with 1 million cells. You're going to back down depending on the hours post-infection you're trying to do. So for example, before starting this, what we did was we plated all of the cells for all to the different time points we were going to use in the future for 0 to 120 hours post-infection, okay? So we just plated them without the virus. At 0 hour, you count them 24, 48, 96, and 120, something like that. And then at that time, you know, okay, so at 48 hours, this is the number of live cells that I will have. At 48 hours post-infection, this is the number of live cells that I will have. And then you use that for your calculation. And you also have to take into consideration what the virus will do to your cells. If the virus will rapidly kill your cells, if you want to do your experiments like earlier on.
Okay.
Because if you wait too long, all your cells are dead and everything is leaked out.
Can we start experiment? Sorry, can we start experiment by like 50% of live cells and 50% for dead cell, or earlier than this time?
Oh, you can do that. Yes, absolutely. Like
Okay.
50% or 75%. Okay. BHK grows really fast. So it's like 48 hours, 72. When they don't have more rooms to grow, they will die. Not because of the virus, just because they don't have enough nutrients and room to grow.
Of course, we need just
Yeah. So
2
that's
days
how you
at least.
interpret.
Yeah.
Yeah. So that's about cell numbers. I would advise that before you even start infection or anything, try to see at different time points post-infection that you hope to use in the future, use that and plate the cells for that amount of time. And then you can know that, oh, they start dying at 48 or at 72, and how many cells you're going to get at those different time points. And you factor that into your infection protocol later in future. Does that answer your question?
Yes.
Okay.
Because
Then-
you didn't mention that in your presentation, so I just want to explain more.
That's okay. Thank
Thank
you.
you, Dalia.
And then the dilution, we have another question about dilution
Yes.
of the antibody.
Yes.
Yeah,
You
so
just follow the manufacturer, or you also do optimization?
Oh, yeah.
Okay.
Yes. You have to do optimization. And sometimes you see papers that say, oh, we used 1 in 200 or 1 in 50. Or
Right.
it doesn't mean that would work for your own
Okay.
cells. Even
So
if
we
it's
need to
the
do
same
2
thing.
experiments before this experiment.
Yeah, once you've established your cell replication, growth, and everything, then you need to establish your dilution too. Like
Yes.
start from the biggest dilution that would really like almost blind you. And
Right.
then you start from the lowest one that you be like, what is this? Yeah, something like
Right.
that. So it's from like, for example, 1 in 100 to 1 in 1,000, something like that, or 1 in 1,000 to 1 in 100 concentration.
Okay.
Yeah.
Thank you. Thank you.
Yeah.
Thank you, Dr. Dalia. And about the question of Dr. Dalia, Dr. Shakira, do you remember I had this issue when I was isolating primary macrophage, then stimulate them to be mature osteoclast. So I had, let's say, hard time to choose the 48 wells or 24 wells plates, and how many cells I have to put per well, 50,000 macrophage per well or 25,000, because the osteoclasts, they are bigger, maybe 15 to 20 times or more than mature macrophage. So I have to standardize like different protocols. Then I pick up one that helped me to finish my experiments. So this is very, important things we have to do before start the real experiments.
Yeah, and that's actually a good point because different cells have different shapes and sizes, right?
Yeah.
It's just like different gumballs of bubble gum or something. They have different shapes and sizes. Maybe 20 will fit in this box and only 10 will fit in this box. So it's different. Okay, so another thing that can help you to jumpstart to think about, okay, so what am I going to start with is go into the literature and kind of see what's out there about the cell you're trying to do. Even though maybe you're working on Brucella and then the cell line is for, I don't know, a virus, even like another kind of bacteria, what you're concerned about is the cell. So you don't have to restrict yourself to a particular publication because it doesn't really talk about your Brucella or something like that.
And you remember like more of the methods, they are like, there is some missing information.
Right. So
like how many rate per minute you have to centrifuge the cells, the incubation time. So we have, I think, standardized on protocols. I think this is the best.
Yeah. And for your calculation of your MOI, multiplicity of infection, and that's really important too, because you want to know how many virus particles you're using to infect at least one cell. So for example, if you're using MOI1, it means that you're infecting one cell with one virus, sort of. And if you don't know the number of cells that are there, how are you going to calculate your MOI? So everything has to be like, you're going to have to troubleshoot, and that's okay. And all of these things, before a paper comes out, it takes about a year or even more than that for some papers. But you just finish reading it in like what, 20 minutes, 30 minutes? A lot of work has gone into that. And like Dr. Omar said, not everything is put in the publication like that, because there's restrictions of how much information or you can put on there because of word count and things like that. And that's what I've done in the past. I've emailed authors of papers and be like, oh, so I saw that you did this, blah, blah, blah. Could you help clarify this method or something like that? And that's why the scientific community is huge. You can ask authors questions. That's why there's transparency. It helps with transparency in research. So it's okay to email people and be like, okay, so I'm having a hard time with this. How did you go about it?
Exactly. Thank you. Please enter question. Anyone has a question to Dr. Shakira?
I'm gonna ask a question before somebody raise your hand. His hand. So do you have, Dr. Shakira, a problem with the autofluorescence? Because when using the tissue sections in pathology, you know, yeah, that we like tissue sections and we do a lot of staining on the tissue sections. So do you have a problem with autofluorescence? I always have a problem with the autofluorescence when I'm staining fluorescent antibody, not chromogenic.
Yeah, so there is another control that I should have added to my list of controls. It's called the endogenous tissue fluorescence something, because yes, there are cells that have this autofluorescence and they can interfere with your signals. But I think with more sophisticated machines of fluorescence microscope like the Keons, it helps you. There's some calculations that you will look at and helps you kind of like subtract autofluorescence signals from your actual signals. Okay, so there's a way. Let me see. Sorry. Where are you? Okay, here. So all of these numbers, there's a way that you can adjust them in the system to help you kind of determine that the signals you're looking at, it's mainly your proteins and not the cell's autofluorescence. So for example, here, it's really clear, the one on this side, right? It's really clear that I'm looking for red signals and they're inside the cell. And what you would tell the computer is, I'm only interested, there's a way you can do it that you can tell the computer that I'm only interested in these red signals that are inside the cell and nothing outside, okay? So that's why all this sophisticated system can help you kind of reduce the signals that your autofluorescence signals from just endogenous protein production by cells.
Yep.
Yeah.
Yeah, and you have mentioned the difference between the direct and indirect method, and you said that it's mainly about cost, not specificity or something like that. So is that the only reason when you're choosing between these two methods?
No,
because I'm not sure. Yeah, because I'm always when I'm using the direct, sometimes it's not working perfect with the tissue, but then when I use indirect, it's get more signal. It's even antibody that doesn't work with the direct, you might sometimes they're gonna work with indirect method. So I'm not sure if you have noticed a similar thing.
Yeah, so they talked about, I didn't really use primary in any of my experiment. I've always used indirect because I read that it amplifies your signal better, you know, so that's why we use indirect, but definitely it's not just about cost, why you choose direct over indirect. It's not about cost, it's faster. And then the thing is, if you're using like a rare protein, it's hard to see primary antibodies. They already have the fluorochrome conjugated one with it that you can use. But I have good experience with indirect method in the amplification of my signals. And in terms of like specificity, you see that the you have the like if you use goat anti-mouse, it means that it would recognize only mouse, only mouse antibodies. So that could also help in like helping to clean up your signals. Another thing that I usually do that I forgot to mention here is you need to block background signals before you start staining. And that would also help you for your autofluorescence that you're experiencing. Sure, so before I start, what I do is I add normal serum. Depending, there are several types that you can use. So you block autofluorescence or like non-specific protein binding with that serum. Then you start. It usually gives you a cleaner signal. Yeah.
Have you? Okay.
Dr. Dalia, she has a question.
Go ahead, Dr. Dalia.
Sorry, Dr. Shakira, to follow up this question about the differences between direct and indirect IFA. So, you know, there is non-specific signal. Sometimes we like find non-specific signal. What do you think? Where is the non-specific signals we can get from the direct or indirect? And also for your experiment, did you get any non-specific signal?
Oh, is that all your questions?
Yes.
Okay, yeah, I was waiting to hear everything. Yes, so did I get non-specific signals? Not really. because I used the blocking step before I started staining my stuff, my cells. And then with the Keon's microscope, I was able to like neglect some signals. There's a way that you can do it where you can tell the program that you're only interested in these kind of signals. So every other thing from like a dead cell, you know, dead cells usually spill things that get bound with antibodies and things like that. So you can program it in a way to tell you that these are the signals that I'm interested in. And then what I did essentially is that I chose the brightness level that I wanted for my signal. So if there's anything that's not as bright as that, I kind of neglect it to help me reduce potential autofluorescence from like dead cells or proteins from dead cells. Yes, so that's what I did. Then your other question was about specificity?
Between the both methods from direct and indirect, what do you think? The direct has more non-specific or indirect?
So my experience again is with the indirect, and I didn't really have any problems with that. But the direct,
I agree with you because you do have two types of antibodies.
Exactly.
Yes.
Exactly. So, and yeah, I would go with indirect, but some people will like, well, if you have the money and the antibody is available and it's conjugated, maybe direct is better because it's faster and you can just like move on. But when it comes to specificity, I think that you have
direct is more specific than
yeah, because you have just that one. With the indirect, it's possible, okay, because what your GOAT anti-mouse is recognizing is any mouse protein. So it's possible that they bind some proteins that are just, you know, mouse protein. Do you understand what I'm saying? So
yes.
So that would kind of like add to your signal, because when you're trying to direct this antibody against mouse, this
is why we have controls. This is the reason, why we have controls. Yes.
For every plate that I used for the experiment,
control. Yes.
Exactly. You have your positive control and your different controls. So even if I have I had several cell lines, right? So I'll put like maybe 24 hours post-infection in different wells, and then this separate well will be for my controls. And I always either use the far end wells or the lowest, so there's no spillover while I'm titrating or pipetting or something like that. I don't spill into that. So I usually leave some space between that, the infected wells or my real samples and controls. And then learn to change your tips and learn to change your tips, learn to be very...
I did that on the slide, so it's more difficult than you like played them. So I did it on the slide. So it is more difficult, you know?
Yes.
Yes.
Yes, because they're close, the wells are really close together.
Right.
On the slide, yeah.
Yes. Okay.
I used the slide, I didn't really enjoy it, so I just dumped it, and I used the plate eventually. Because with the slide, you also have to be careful, and that goes back to the number of cells per well too, okay? Sometimes the well, because they're in the slide, they either clump together in the center, or they go to like the corners. And when you're putting your antibody, you just put it in the center, and if it doesn't diffuse all the way, or you have clumped cells in some corners, it can affect your signal too.
Yes.
Yeah, and then so that's why when I was using the slide after plating the cells, before infecting, like I kind of like shake them to make sure that they're all like widespread before I put them in the incubator, because you want to put them in the incubator when it's all spread so they can attach to the cells and keep, you know, growing. But if you don't do that, you just put the cells and put it in the incubator, it could just be a clump of growing cells by the time you're ready.
Yeah.
Does that make sense?
Yes.
Yeah. Okay.
Thank you.
Yeah.
Dr. Sherry, I have a question, Dr. Shakira. when you add the DAPI at the last step, right? To
Mhm.
stain the nuclei.
Mhm.
After that, you put the PBS or formaldehyde, like to keep the cells wet. So what you have chosen?
So I use the DAPI from a manufacturer. You don't have to use that. Just put the DAPI
Okay.
and then that's it. Always remember, yes, I forgot to talk about this. When you're done with your staining and you have your DAPI, your final step, if you're not able to image it that day, you need to keep them wrapped in foil as much as possible, so you don't have like loss of your fluorochrome, exposure to light. Yeah, that's it. That's the word. Avoid exposure to light.
Yeah. So I
Yeah.
mean, you have also, I mean, you add DAPI only, right? And that's it.
Yeah.
With
Yes,
the PBS.
and that's it. No. So the DAPI it's kind of like this oily thing that it doesn't dry up your cells. It has like a finishing touch that I can't remember right now.
Okay.
That was the only thing that I put, and then I cover it up with foil.
That's it.
And if I'm too tired that day, I can image the second day.
Means you haven't like washed them after DAPI and put PBS, and no, you haven't done. Okay.
No. I mean, I was using intracellular proteins, so my cells were already fixed and permeabilized. So yeah, I don't have to fix them again because they were fixed earlier on because I was using intracellular protein.
And what kind of solution, like fixative?
Oh,
Yeah.
I used the
Formaldehyde?
No. Yeah, I think formaldehyde was in there, but I used a kit from the manufacturer.
Okay.
Yes, I used the kit. The kit has its own. In fact, it's really very easy. It has everything. Like you have your fixation buffer, permeabilization buffer. You even have like blocking solution in that same kit. So the only thing I needed was my antibody and diluent and things like that. Yeah.
Nice.
Yeah. And remember also, if you're doing multiple proteins
Labeling
from the cell, maybe one on the membrane and one intracellularly, make sure that you stain the one on the membrane first, then you fix, permeabilize, and then stain with the other one that's inside the cell.
Oh, this is very like nice step.
Yeah, that's kind of like the sequence that I go through. And if you're staining multiple proteins, you have to take into consideration the fluorochromes. And this is where autofluorescence and fluorochromes overlap between the emission spectra, okay? So you have to be careful, like you're not using maybe like red and orange together, that kind of like looks the same. And then you have to be careful about the antibodies. And this is where it gets complicated when you're using indirect method, okay? For example, let's say these are two epitopes that we're looking for. This one from a mouse cell. This one is mouse anti-X, okay? So now you're using goat anti-mouse, right? You're done, okay? Yeah. But this one is also mouse, right? So you wanna use maybe rat anti-X for what this protein is. You cannot use goat anti that. So that's how you have to be very, very careful and plan your experiment right. If you're staining for multiple proteins. Exactly. Okay. Always remember what host species that your antibody is made from. So this goat anti-mouse means that it was an antibody from the host species is a goat, but the antibody recognizes mouse.
Mouse. Yeah.
Does that make sense? Yeah.
Makes sense. Exactly.
Yeah, it's complicated, but
no, it's very clear.
It is complicated, but you're making it more clear.
I have another question, Dr. Adetunji. Have you done confocal with these infected cells?
No, but yeah, I didn't do it for this one, but the Keon's microscope that I used can do for confocal, but it was not. Yeah.
You were lucky to have that machine, right?
Yeah.
So we have to also be like remember, if I'm using my, like, let's say Olympus fluorescent microscopy, I have to be sure the filters that I have, right? Like FITC, TRITC, DAPI, all these like colors. So
Yes.
it's important before like I'm ordering the antibodies.
Yes, you have to know the like you said.
Yeah.
Another thing is that even if you don't have the fancy microscope, because we've done it before, right, Dr. Omar,
Yeah.
where we don't have that kiosk, right?
Yeah.
Another thing you can use to like quantify your signal will be image J.
Yeah.
And I think it's widely available. So once you get your images, you can put them in image J and use that to quantify like how dark or how intense your signals are, and put that in a statistical table or graph.
Yeah. The same way we're using the clicker, right? That machine to count the cells before.
Yeah.
Thank you for the questions. Any question, please, to Dr. Shakira?
I don't see any questions. So
No, nobody.
I think Dr. Shakira done very well. So everybody had enough information. Thank you very much, Dr. Shakira, for the information that you have presented.